We usually think of human DNA as a rigid architectural blueprint, a strict set of physical instructions. But looked at through information theory, the genome is actually a highly compressed, multi-million-year historical archive of ancestral survival data. From a structural standpoint, this means the human genome operates like a pre-trained machine learning foundation model. Map the AI pipeline onto biology. Natural selection becomes pre-training. Left, AI parses text using gradient descent to encode weights. Right, species navigate environments using natural selection to encode biological code. Both share a strict loss function. AI predicts text tokens, biology survives to reproduce. At the end of these pre-training phases, both systems enter active deployment, and neither one starts from scratch. They arrive with massive preloaded assumptions about exactly how their operating environment works. Human cognition does not begin as a blank slate. Our brains are initialized by deep evolutionary data, acting as structural inductive priors. In biological literature, neuroscientist Tony Zador formalizes this concept as the genomic bottleneck. The bottleneck is a physical data constraint. Biological organisms cannot pass down uncompressed, complete life experiences to the next generation. An ancestor can't hand off a specific memory of a predator or a map of a watering hole. Instead, millions of years of ancestral trial and error are forced through a compression channel. Infinite environmental interactions distill strictly into a tiny sequence of discrete genetic bases. At birth, this sequence blooms outward, initializing the brain's hardware, topology, and the neural connectome's synaptic predispositions. These structural predispositions manifest as innate priors. It is why newborns arrive already expecting physics to exist. They exhibit built-in assumptions about gravity, object permanence, and face recognition within hours of birth. This structural mapping is elegant, but if we interrogate the actual optimization mechanics of biology, we run into strict mathematical boundaries where the AI analogy shatters entirely. The divergence between artificial networks and living tissue happens at the optimization layer. It comes down to exactly how each system updates its code. Artificial backpropagation relies on a top-down, vector-directed process. Using matrix derivatives and the chain rule from calculus, the system computes exactly how wrong an output is and issues a precise directional command to update all weights simultaneously. As pioneers like Geoffrey Hinton have pointed out, biological brains simply do not have the architecture to run backpropagation. In fact, there is absolutely no biological mechanism to compute global derivatives and pass specific error vectors backward through synapses. Natural selection operates as a blind scalar filter. The environment cannot send a directional instruction back into the genome, saying, shift the metabolic gene by exactly 3%. It provides a binary signal, 0 for death or 1 for survival. Without calculus to guide it, biological updates rely entirely on a random walk of stochastic mutations. It is an incredibly inefficient process of trial and error driven by genetic drift. AI optimizes over a differentiable continuum, allowing for microscopic fractional tweaks. Genomes are built on a discrete digital code of A, C, T, and G. You cannot smoothly slide a base pair. A single letter mutation can cause a catastrophic binary failure, like completely misfolding a protein. Biology also suffers from a phenotypic proxy constraint. AI optimizers access their neural weights directly. Evolution only selects based on physical bodies exposed to environmental noise. A highly advantageous gene can be permanently deleted from the data pool simply because the animal carrying it unluckily stepped under a falling rock. Because biological evolution is mathematically incapable of functioning like artificial gradient descent, we have to reassess how life actually fine-tunes its data. This brings us to the second half of the analogy, the assumption that epigenetics acts as a transgenerational fine-tuning layer, similar to snapping a LoRA adapter onto a neural network. To evaluate this, we look at the nested memory systems framework. This model positions cognitive processes on top of a multi-tiered stack of biological memory, ranging from deep evolutionary history at the bottom to externalized culture at the top. In this stack, the epigenetic layer theoretically acts as a dynamic mid-term cache. It sits between deep genetic hardware and lifetime learning, adjusting gene expression based on the recent environmental stressors of your immediate ancestors. The biological reality is that durable, transgenerational epigenetic inheritance in mammals is highly contested and mechanically limited. The primary barrier is gametogenesis. During the formation of sperm and egg cells, the vast majority of these chemical epigenetic tags are explicitly erased. The cache is wiped clean. We do have historical cohort studies pointing to transgenerational trauma, most notably the data stemming from the Dutch hunger winter in 1945. But while these findings are statistically suggestive, the mechanisms of long-term epigenetic transfer remain scientifically unsettled. If the epigenetic fine-tuning layer is largely erased between generations, a strictly top-down AI-style deployment model is biologically incomplete. Without a durable transgenerational cache to pass notes across time, we face a mechanical architecture problem. How does an individual's lifetime learning actually interface with the deep evolutionary code? The biological mechanism that solves this is Waddington's genetic assimilation, operating alongside the Baldwin effect. Traditional frameworks assume a one-directional relationship. They argue that deep phylogenetic hardware strictly dictates shallow ontogenetic learning. The code sets the boundaries, and the organism learns within them. The Baldwin effect provides the missing feedback arrow, creating a path back up the biological hierarchy. When an organism makes real-time, localized adaptations to survive its immediate environment, that lifetime learning actively biases the phenotypic traits that natural selection eventually hardwires into the species' DNA. The flow of biological information is a loop. Individual lifetime learning shapes the long-term evolutionary data stream. When we look at the highest layer of this loop, synaptic plasticity and the speed of individual human cognition, we see rapid, real-time networking. This learning occurs within the structural inductive priors initialized by the genomic bottleneck. The genome truly is a pre-trained evolutionary foundation, but it gets optimized through a discrete biological feedback loop of survival, not the continuous math of gradient descent. An artificial intelligence model optimizes data inside the isolated, sterile vacuum of a digital sandbox. Biology requires physical stakes. Human consciousness is neither a blank slate nor a static computational program. It is the continuous, real-time execution of an ancient, multi-tiered biological data stream.